Hello, good afternoon, everyone. Let's get started. We will do a quick recap of the lecture 4 stuff. And then I want to start on lecture 5 today as well. And of course, you have your lab that is going to be due on Wednesday. So there will be no class, no Zoom class on Wednesday. Okay. I left my charger for the laptop at work, at school. And so I'm using another computer today, and this one doesn't have a camera, which is why I'm off camera. But that's okay. You don't need to see me. You see me twice a week already. Okay, so this is a quick summary. Right? Summary of membrane.
Vincent, I have a quick question before we start, so I
Sure.
don't lose so
Yeah.
much. Okay, so in the study guide, so the definition for diffusion
Mhm.
says like movement of solvents.
Yeah. Moving a solute, you mean?
Yeah,
Solute?
solute. Yeah, moving of solute from area of high concentration to low concentration, right?
Yeah. Yeah.
Okay, but is also the same thing as movement of molecules.
Yeah, I mean, like the solute is basically the stuff that is in a solution, right? So those molecules, substances, movement of particles, all those things are okay.
Okay, thank you.
Yeah, no problem. All right, so if you are copying along with me, you should put your paper sideways and start in the middle because I'm gonna do a flow chart like this. Okay, there's gonna be a bunch of stuff. Okay, it's gonna be something like that. Okay, so just plan ahead with your space so you don't run out in the middle of the note. Okay, so let's start in the middle. We did this already. Okay, like I'm just going through with you again to help you remember them. Okay, so membrane transport, which is just to move things across the cell membrane. And we generally can separate them into two categories. Okay, so here we have our passive transport, for example. Passive transport. Here's the passive transport. And we have a few examples. The first one was just brought up. It's diffusion, simple diffusion. Simple diffusion. All right. For those of you just joining us, we are just doing a recap, a summary of the transports. Okay. So simple diffusion. This is ideal for, we can say best. Okay, best for molecules that are small. Okay. So things like H2O, that's pretty small. Non-polar, things like CO2, O2, these are non-polar. Non-polar means they don't have they do not have uneven distribution of electrons, and there's no slightly positive or slightly negative part. By the way, I found out many people didn't know this, but if you go on the Zoom ribbon and click on more, there is an option to turn on caption. Okay, so some people find it difficult to follow what I'm saying. You can turn on the caption. It will basically transcribe it live as I'm speaking. Okay, so you might find that a helpful feature. Okay, go to the ribbon, click on more, and then click on caption. So best for molecules that are small, non-polar, as well as things that are hydrophobic. Okay, so like steroid, right? So these things, they use simple diffusion. And then we talked about osmosis. Osmosis, which is basically diffusion of water, right? It's a type of simple diffusion, but specifically we're talking about water. And so the thing you should review would be, what happens to water in a hypertonic, isotonic, and hypotonic environment. Right? Review to yourself how water is going to move, in different direction, causing the cell volume to change. Right? That's the note that we took last class, right? Okay. And sometimes things are too big and they're not able to cross the membrane on their own. And so they need to rely on the help of um, membrane proteins, right? So this is called facilitated diffusion. It's still passive transport, okay? Which, sorry, I should have put it down here. Passive transport is no ATP is needed, and it's going from high concentration to low concentration, right? So facilitated diffusion is best for large or charged particle molecules. Doesn't really matter. Okay. It requires the help of a membrane protein. And can anybody remind me what the two types of facilitated diffusions are? Does anybody remember? It's okay to look at your notes if you need to.
Channel and carrier.
That's right. Very good. Thank you. So there are the channel proteins, and then there are the carrier proteins, right? So let's do channel protein first. Okay, so the channel proteins are the ones that create a tunnel. Here is a channel protein. And then there's a tunnel in the middle. And that is ideal for charged particles to go through. Okay, the tunnel shields the ions from the hydrophobic tails of the membrane and allows them to pass through. Okay. On the other hand, we have, I mean, for the exact definition and description, you can look at the PowerPoints, right? And then for the other one, it was a carrier protein. So this one, it's like a revolving door, right? The protein actually changes shape, not the shape of the molecule that it's transporting, but the protein itself changes shape, and in doing so, it transports the material over. So it grabs onto this big thing, and then the protein changes shape, and now it opens on the inside, and that's how you move. That's how you move the molecule across. So this is useful for like big molecules, like glucose. Okay. So this is best for large molecules like glucose and charged particles like Na plus, K plus, all that stuff. So that these are the passive transport, okay? Give you a moment to catch up.
Vincent, is channel protein Na plus an example of channel proteins?
No, Na plus is an example of something that is transported
Okay,
through the
okay.
channel protein. Yeah. Okay, good question. All right, so we're gonna go back to here, and we're gonna have this active transport now, right? Active transport, which requires ATP, and this is going from low concentration to high concentration, right? And the first type we talked about was a protein pump. All right, so this thing again needs a membrane protein, and I mean it sounds very similar to an active transport description, but it uses ATP to transport molecules, particles against their concentration gradient. Okay, it goes from low to high, uphill, against concentration gradient. So we had two examples in class. I mean, I didn't really talk too much about them. One of them is a proton pump. Proton pump. All the examples are in red. Okay, proton pumps. Protons are H plus pumps in the stomach, right? That's what makes your acid acidic in the stomach. Right? So you need to constantly pump H plus into it. Like if you think about this as the stomach, you learn this next semester. That's your stomach. The cell and the linings, they constantly put H plus into the stomach, and that makes the acid. The other example I gave you was the sodium potassium pump. Okay. This one we I mean I didn't give you too many details about it. But let's say this is the inside and this is the outside. Okay, well, what the pump does is it will pump 3 Na plus outside, and it will bring 2 potassium in. So you keep on doing this, there's going to be high sodium outside and high potassium inside. But you keep on putting more there, which is why it is an example of a protein pump. You are pumping them against their concentration gradient from low to high. All right. Give you a moment to catch up.
Please explain the sodium potassium pumps again, like the simple one, like putting in sodium or.
Yep. So let me just pull this out here. Okay. The sodium potassium pump will pump 3 sodium out of the cell. So over time, let me find a bigger pen here. So over time, you will have a lot of sodium outside. You understand? And then not a lot, right? So then when you pump sodium out, you are taking it from an area of low concentration to an area of high concentration, right? Similarly, for the potassium, because you keep on bringing them in, you're going to have a lot inside, right? So this is high potassium and then low potassium outside. So when you pump the potassium in, you are taking it from area of low concentration to an area of high concentration. That's why you need to have a pump, right? To go against the concentration gradient. Just like pumping water to the top of a water tower. It takes energy, it takes electricity. Is that a little bit better?
Yes, thanks.
Okay. Anyone else have any other questions? Okay. And then we have bulk transport. Bulk transport. This is like moving large quantity transport of bulky things or a lot of things. Okay. Bulk, the word bulk means really big, right? Bulky. Something is really big. Or when you say to do it in bulk, that means you do it in large quantity, a lot of it. You know, people go to Costco and they buy in bulk, buy a lot of the same item, right? So in this case, bulk transport, you either are moving something super big, like a bacteria that's really big, or you're moving a lot of something, even if they're small. But if you need to take a lot of them in, that is considered as bulk transport. And there are 2 types here. Okay, we have the exocytosis. That is moving things out of the cell. Okay. So releasing things, whatever the things are, out of the cell. Okay, so maybe take a moment and see if you understand what is it that I'm drawing. So like this is cell membrane. Okay, just watch first. And then we have a vesicle here. Okay, so inside this vesicle, we have some stuff, whatever it is. Could be proteins, could be, particles, doesn't matter. Okay, so you want to release this out of the cell. It's going to move towards the cell membrane, and then it's going to touch the cell membrane. It's going to touch the cell membrane. And then it's going to start fusing with the cell membrane. It's going to start fusing with the cell membrane. And eventually it's going to become part of the cell membrane. And all this stuff is going to get out of the cell. You understand? Right. That's how you read the diagram. This is, of course, cytoplasm. Okay, so this is exocytosis. You want to be getting some things out of the cell, into the surrounding. Do you understand this? Let me know if there are any questions. Okay, so opposite of exocytosis is endocytosis. Endocytosis is to bring things into the cell. Okay. And what are the three types?
Phagocytosis, pinocytosis receptor.
That's right. Those are the kind, right? So we have three different types. We almost done with the chart, okay? We are almost done. We have phagocytosis, pinocytosis, and then receptor mediated endocytosis, right? I'll let you write down first, and then I will explain. Okay. Phagocytosis means cell eating, right? So e.g., oops, that's too thick. E.g., it is like if you eat a bacteria. Bacterium is a singular. Like your white cells eating the bacteria. Okay. Pinocytosis would be cell drinking. Right. So bringing in extracellular fluid from the outside. And for receptor-mediated endocytosis, it's probably best to illustrate it with a diagram. Okay, so we have some receptors on the surface, and let's say in the surroundings we have these red circle balls. Okay, and then we have some green triangles. So which one are we going to be able to bring in? Only the red ones, right? In order to bring whatever in, it has to match the receptor. You understand?
Yes.
All right. Thank you. So that's it. That's a summary, guys. That's everything we learned about transport. Okay. It's a good idea to rewrite this maybe, right? Have it next to you when you do the quiz next week. And again, I want to emphasize to you, it's okay to have notes next to you. All right, I don't want you to not do it and not be able to use that advantage. Okay, for my course, it's okay. Right, but don't have like your whole binder. Like I said, it's hard to dig through everything and find the answer on the spot. Create a summary sheet. Summary sheet. Right, something like this is a summary sheet. Right, then you can see very quickly. If I ask you which of the following is an example of active transport, you can see it right away. Right, protein pump, exocytosis, phagocytosis, so on and so forth. Right. Talking about the quiz, it is going to be next Monday. Okay, your quiz number 2. I'll post this information later on. Okay, but quiz number 2 is going to be Monday next week. What is that? That would be February. Today is the 5th, so 12th, right? That's gonna be your next quiz. 10%, lecture 3, lecture 4. Okay, we do have class at our regular time. It will be available from 6 a.m. to 11:59 p.m. All assessment in this course is available, including the test, is available from 6 to 11:59 on their day. Okay. So again, no class on Wednesday. Use it to do your lab. Some of you already finished your lab, okay? Amazing. You use the time on the weekend to do it. That's great. Okay, but if you haven't done it yet, the lab is available to you until Wednesday, end of Wednesday. Okay, like 11:59. Oh, actually, I think it's 6. Let me just double check. Let me just double check what I said here. Until, let me see. Just want to be sure. Yeah, 11:59. Okay, and it's going to be available until the end of Wednesday. Okay, so please be sure to complete that. Any other questions before I move on to lecture 5? No, everybody's happy.
Professor,
Go ahead.
can
Yeah.
you please a little bit the small size of this page? I need to take a photo.
Oh yeah, I'll post it online though afterwards. It's
Okay,
probably
thank
easier. Yeah, because if I zoom out too much, it's like you can't see, right?
Okay,
Yeah,
thank you so
but
much.
I'll post it.
Thank you.
Yeah. Okay. All right. So like I said, last time I looked at the class, average, it was pretty good. So just keep doing what you're doing. Okay. If you need help, let me know. I'll try my best to help you. Okay. All right. So lecture 5 is really hard. I'm not gonna lie. Okay. So this is lecture 5 notes. Okay. If I go through the PowerPoints, like I don't think it's too effective. Okay, like I've been doing this for like 15 years, and then when I do use the PowerPoint and just teach you through the slides, people don't really get it because it's such an abstract concept. And so I'll try to teach it to you through the notes first, and then next week, right, maybe we'll go through some of the important slides. All right, so get some new paper. Some colors would be nice, and then I'll teach you what I can. So this is called, oops, forgot to change the thickness. This is called metabolism. Metabolism. So we learned about metabolism, right? A little bit. We know it is all the reactions that are happening in the body, and some of it is going to be catabolic, where you break things down. Some of it is going to be anabolic, where you have to build things. But the most important metabolic process in the body, arguably, is cellular respiration. Okay, cellular respiration. And it goes like this. Okay, we'll write down the word equation first. This is word equation. I'm not sure if you learned chemical equation and word equation in chemistry yet, but we have the glucose plus oxygen, and then it's gonna react with each other to produce carbon dioxide plus water and ATP, okay, which you might remember is the currency for energy in a cell. We use ATP for different things, for chemical reaction. We use it for movements, right? We call it mechanical work in the slides. We can use it for transport, right? But to make it, this is the main method of making ATP. Okay. Now we can write this as a chemical equation as well. So the difference between chemical equation and word equation is that word equations are word, and chemical equation is the chemical formula for those words. So by now, you probably know glucose is C6H12O6. So that's glucose. And when you react it with oxygen, oxygen is O2. I think in your chemistry, you learned that some elements are always doublets, right? Like oxygen, nitrogen, hydrogen, right? So on and so forth, right? That's going to produce carbon dioxide. Does anybody know the chemical formula for carbon dioxide?
CO2.
Yep, very good. CO2 plus water and ATP. Okay. Now we have to balance the equation. Did you guys learn balancing equation yet in chem?
No, but I think it's next.
Next, okay, well, that's okay. You just have to trust me then. Okay, the point of balancing equation is to make sure that you have the same number of atoms on both sides. So if you put a 6 here and a 6 here and a 6 here, then everything would be balanced out. Right, so like for example, I have 6 carbon on both sides. Okay, like 6 times 2 is 12 hydrogen, and then I have 12 hydrogen, so on and so forth. Now you might wonder, hey, how come there's ATP on the right and no ATP on the left? This is just like the energy that you're able to harness from the equation here, right? Okay, so it's not part of the atoms. And you can make up to 36 ATPs. All right. Now I want you to leave some space here. Okay, leave like 3 or 4 rows. We're gonna add more things to it. But I want you to know cellular respiration is a redox reaction. Okay? It's a redox reaction. I'll explain what it means, okay? In a redox reaction, electrons are transferred from one reactant to another. Reactants are the ingredients, okay? They are the things that are on the left side of an equation. So electrons are going to be transferred from one reactant to another reactant. Okay, so there are two things here. Okay. We have reduction. A molecule is reduced when it gains electron. And you can gain more than one, it's fine. So that's reduction. That's where the in the red, I guess that's where the red comes from in redox. And the opposite of reduction is oxidation. Oxidation. Okay, so a molecule, and I guess it doesn't have to be a molecule, it could be like a particle, right? A thing, okay? A thing is oxidized when it loses electrons. Okay, so you can remember it like this. You can remember like oil rig. Oxidation is lost of electron. Reduction is gain of electron. Okay, oil rig. You know, like in the ocean, and then they have the thing, right, to take out the oil. That's an oil rig, right? Okay. So you have to understand in cellular respiration, the glucose is the one that is going to be oxidized. And it's going to be oxidized into carbon dioxide. In other words, in order to harness the energy from glucose, we're going to be ripping electrons from glucose. We're going to be ripping high energy electron from glucose. And eventually, those electrons are going to be picked up by oxygen. So the oxygen will gain the electrons and it will become water. So this is reduced. So the electrons are going to basically transfer from glucose to oxygen. Okay, so that's like a lot of information right there. Any questions? Anything you need me to clarify? Please let me know.
Yes, the ATP.
Okay, yeah, what of it?
Yeah, so does that mean the energy needed? Okay, so since cellular respiration is the way most organisms obtain energy from glucose, is the energy the ATP? Like, do we run on ATP as like as a living organism?
Yes, so our cell runs on ATP.
Okay,
and
okay.
remember last class, and you guys don't have to copy this if you don't want to, okay? You already have this in your slides, okay? So like we had the, remember the A, T, and then like a P, P, P?
Yeah.
Right, and I said like the last bond is really strong, right?
Strong.
Yeah.
Yes.
So like when you take it off, then now it
The
becomes
ATP.
ADP, right?
Yes.
So in order to recharge this molecule, in order to put that P back on,
Yeah. Okay.
I need a source of energy, right? So a source of that energy is from burning the glucose here.
Glucose. Oh,
Yeah. So
okay.
by doing this reaction, by ripping the high energy electron from it, it's gonna release the energy. But we must capture it If we don't, it's going to be lost forever. And that's where the ADP comes in, captures it, and then it becomes the ATP again.
ATP. Okay, thank you.
Yeah. Does anyone else have any questions about this? Reduction means gain electron. That's right. So if something picks up the electron, as is the case with oxygen here, we say it has been reduced. Okay, so a redox reaction, I forgot to highlight here. A redox reaction is essentially a reaction that involves both reduction as well as oxidation. Okay. Okay, these are good questions. Just want to make sure everybody is still okay with me. All right, fantastic. Let's move on. Now, even though this reaction looks like it's one step, right? It's just, glucose reacting with oxygen, and then you get the products. But actually, we have to do it in many, many mini steps. And we want to extract the energy a little bit at a time. It's kind of like your car. Right? You have a full tank of gasoline. Rather than, lighting a match and throw it in the gas tank, that's gonna burn all the fuel all at once. It's very powerful, the explosion, but it's not very useful explosion. Okay, you use it all up, all the gasoline up in one go, right? It doesn't get the car very far. Whereas if you do small controlled explosion, which is what's happening when you're driving your car, right? That explosion moved the car in a meaningful way, in a productive way, right? So we don't want to burn the glucose in a single step. Okay, like I can actually show you, like in an in-person environment, I could show you how violent the burning of glucose is. You can Google that up yourself on YouTube. Okay, like you can see people burning the glucose and it just goes in flame. Very, very powerful. But we don't want that. Okay, number one, it's gonna burn our cells, right? And the temperature is gonna be too high. And number two, like the explosion of the gas tank, it's not really meaningful. It's not very productive, right? So we break this equation down into different stages and in many smaller steps. So I'm going to give you an overview of what that is like. Okay. An overview of cellular respiration. So this is the cell. Oh, I like my original better. There we go. There's the cell. This is not to scale at all. It doesn't matter. Here is the thing. Hold on a second. Can someone tell me what am I drawing, please? Please tell me you know.
Mitochondria.
Oh, thank you. That makes me feel better. To know that you guys know what I'm teaching you. Okay, so that's the mitochondria, and it's not surprising I'm bringing up the mitochondria, right? Because that's where the energy production occurs, right? So outside here, of course, is the cytoplasm, right? All right. Overview of cellular respiration. So there are 4 stages. 4 stages. Okay. First one is called glycolysis. Don't worry about what happens in each stage and what the word means and all that. We're gonna talk about it later. All right. For now, just gonna write them down. Okay. Stage 1 is glycolysis, and it happens right here in the cytoplasm. Okay. Number 2, we have something called pyruvate oxidation. I know they're really big names and confusing, but they're just names for now. We don't really care what exactly do they mean? And number 3, we have the Krebs cycle. Krebs is a person's name, probably named after the person who helped discover this cycle. And number 2 and 3 happens here. Can someone tell me what that location is called?
The matrix.
The matrix, exactly. Number 2 and 3 happens in the matrix. Okay. We'll write these down later on. But if you want to add it to your notes, more than welcome to. And finally, we have the last stage, which happens right here. And that is the cristae, right? C-R-I-S-T-A-E, the cristae. And that step is called the electron transport chain, or ETC for short. Okay. So there are 4 stages for cellular respiration. Like, glucose does not become carbon dioxide in one go. It happens in stages, and within each stage, there are multiple steps. Like in glycolysis, there are actually 10 steps. Okay. And then 3 steps in pyruvate oxidation, so on and so forth. There are many steps that are involved. And you harness the energy a little bit at a time. Okay. Questions?
Yes.
Mm-hmm.
So the step 4, which takes place in the cristae,
Yes.
and according to the notes, it's the inner membrane to
That's right. The inner membrane that
okay.
folds up, right?
Yeah.
That's
To
right.
increase surface area. So is it like on the increased surface area or where?
Yeah, it's as you will see, it's right in the membrane.
Oh,
Okay.
okay, in the
Yeah,
membrane.
yeah. So like, the membrane is gonna be just like the membrane we drew. Okay, and it's gonna happen like within the membrane.
Okay.
So when I say the cristae increases the surface area, because of the folding, like the wavy things that I drew, you have more membrane available, right, to do this.
Okay.
Yeah.
Okay.
All right. Now I want to draw a line here to separate the first 3 steps from the last step. Okay. And this is the theme that I want you to remember as we continue with the lesson. Okay. What's going to be happening is that, let me choose a nice color here. What we're trying to do here is we are oxidizing glucose, right? What does that mean? Oxidizing glucose means we are, remember, right? Oil rig. Oxidizing is losing. So when we oxidizing glucose over here, we are trying to rip electrons from the glucose. So all these high energy electrons are going to be coming out of glucose along the first 3 steps. Okay, so we have high energy electrons being released as glucose is oxidized. Now, I would, this sounds like a strange thing for a teacher to say, but I would advise you not to look this up on the internet and try to read it up, okay? I am simplifying it substantially so that it's appropriate for our program. Okay? If you go read this stuff online, you're going to get overwhelmed with details that you don't have to know. Okay? If you don't understand it, just watch the recording of this lecture and try to, have another listen. That should be enough. Okay? And, ask me questions. Okay, don't
Wait,
look
I
at it.
have a question. So, all these things is happening here is just ionic compound, right?
What do you? Oh, okay. I see what you mean.
Because
Yeah, yeah, yeah. I try not to
because
bring the.
they are
Mhm.
transferring electron, right?
Right. Okay. So I try not to bring that up earlier. I understand where you're coming from. You were saying we learned, earlier in this course and probably in chemistry that ionic bond is formed between, say, sodium and chloride, and there's a transfer of electron, right? So you're asking me,
Yes.
is this ionic? Well, no. It turns out the ionic bond, that the thing you were talking about, that's another example of a redox reaction. But not all redox reactions have to result in an ionic bond. In this case, we're not talking about ionic bond. We are talking about, literally taking the electron from the glucose and then transferring it onto oxygen. And in the process, very importantly, in the process, there is no bond that is being formed between the glucose and the oxygen. Okay? This
Okay.
is literally like, me passing a ball to you, throwing it to you. Okay? The ball being the electron. And then I have no connection between you and I. Okay? I'm just like throwing the
Okay.
ball to you. All right,
Thank you.
but good point. Okay, these are good questions. It shows that you guys are really thinking about this. All right, so these high energy electrons are being released as glucose is being oxidized. Okay, if we don't capture these electrons, they're gonna go all over the place, and then we're gonna lose the energy. So we have to capture them. Okay, but we are not gonna be capturing them with ADP. It doesn't work like that. Not yet, anyways. Okay, we actually, and I'll tell you more about it in a second. But for now, the big idea is that there's going to be something. There's going to be something that takes the electron, and then we're going to be transporting them to the ETC. Okay, to the ETC, and we are going to be using those electrons at the last step, at the last stage, to make ATP. So let me write that down for you. Okay, high energy electron. Now, from this point on, whenever I say electron, it's understood that it is high energy. Okay, I don't want to keep writing high energy. Okay? So high energy electron, that means electron. Okay, released from step 1, 2, and 3 are captured by electron carrier. Here, I'll write it in red, by electron carrier, which transport them to step 4. To step 4. At the ETC, the high energy electron are used to create ATP. Okay. So let me give you 2 examples of energy carrier. In animal cell, there are 2 types of energy of what did I call them? Did I call them? Oh, electron. Yeah, sorry. Same thing, electron carrier, energy carrier. Carriers. Right? They are called NAD plus and FAD. Never mind what they stand for. It doesn't really matter. Okay? What matters is they are energy carrier. Okay? So let me try to show you this. Don't write it down for yet. Okay? Just try to understand it first. And then you can copy it down. Right? So in step 1, 2, 3, you're gonna release the high energy electron, right? High energy electron, like this. So then the high energy electron is gonna combine with, oh, I lost all my colors, hold on. It's gonna combine with NAD plus, right? As well as FAD. I need more colors. There we go. And when that happens, the NAD plus becomes what's called NADH. Okay, again, never mind where the H came from. It's just around, okay? I don't want to give you too much information to overburden you. And then the FAD is going to become FADH2. Okay, and then these guys will take the electron to step 4. Okay, and then the electron will get off. And in the process, you're going to have NAD plus again and FAD. And then they're going to go back to the first 3 steps, and then this repeats. I give you an analogy to help you understand, huh? Let's say this is the airport. Okay, airport. Airplane, okay? There we go, airport. Right. So like we have different hotels in the city, right? Hotel. Okay? And then there are passengers. The passengers are the electrons, okay? So then there is going to be a taxi. A taxi will pick up the passenger, okay? That's like picking it from like hotel number 1. Okay? It picks it up, and then it drives them to the airport. The guy's going to get off, and the taxi goes back. Oops. The taxi goes back, right, to 1, 2, or 3, right? Do you understand that? Did you guys see the thing move? I actually don't know. Like sometimes I don't think it shows up sometimes. Let me see.
Yes,
Oh,
yes,
it
it
did?
did.
Okay, great. There's like a really low tech animation there, right? But you know, you've seen taxi before when the people is in the car, like there's a light on top that lights up, right? Not the Uber, but then like, you know, the traditional taxi says occupied, right? So that's what this is. Right? Like when you have the NADH, with the H is occupy. It has the passenger. It has the electron in them. You cannot pick it up anymore. So it drives them to the destination, the airport, the ETC, and then it lets the passenger gets off. It lets the electron gets off. And so these electron carriers, they function as shuttles. Right? Shuttles between the first 3 steps and the 4th steps. Okay. So the difference between these two things is like how much energy they carry, which I'll explain in a second. So copy what you need to, and please ask questions. I know this is not too straightforward. It can be confusing. So let me know if I can make it clearer.
Vincent, for 1 and 2 and 3, the differences is just their location, right?
The location, and as you will see later on, what is happening in those steps are also different. But we will not learn it today, probably next week, okay?
Okay, thank you.
Yeah. Okay. Let's take a closer look. I hope you're using color as well, to to help you emphasize certain things, right? So each NAD, each NADH carries enough energy to make 3 ATPs. And again, you just have to trust me. Right? Like it's just the way it is. Like I can explain to you, but then like it's not gonna really help the situation. Okay, like it's just gonna make it unnecessarily complicated. And each FADH2 carries enough energy to make 2 ATPs. Okay, so they have less energy. All right.
Excuse me, Professor.
Yeah.
The FADH2 is the second process of NADH.
Oh, no, they are like different kinds of carriers. So in animal cells, there are two types of electron carriers, right? There's NAD plus and FAD. Think of them as like, like a taxi versus like a SUV, I suppose, right? They are both cars. They both can transport people, but the capacity is different. Like you can fit more people in one than the other. Right? So in this case, you can fit more energy into NAD than you could for FAD. That's the difference.
The energy being the electrons.
Electrons, that's right.
Okay.
We are using them synonymously in this case because we ultimately use the electron's energy to make the ATP, right? So that's why like sometimes I use them interchangeably. Okay. So I will teach you how to make the ATP later, but for now, just remember this fact. Let's try an example. Let's say you have how many ATP can be generated? That's a G. Generated from 10 NADH and 5 FADH2. All right.
That's 30 and 10, right?
That's right.
Okay.
Okay, so like this one, right? This one, I times 3 because each one can make 3. So that's 30. This guy, I times 2. So that's 10 ATP. So the answer is 30 plus 10, 40 ATP. Any questions about this calculation? Okay, well then, let me ask you a few poll questions. We are almost done, okay? So I'm just gonna skip the break and finish the note, and then I'll let you go, you know, a little bit earlier, to, work on your labs and whatnot.
Professor,
Yeah.
excuse me. Why you times, for example, multiply in 3 and the other one you multiply in 2?
Okay,
Can you
so
explain?
that's yeah, sure. That's based on this here, right? So like you want to highlight this fact right here. This is important. Over here. right? So this part here,
Mhm.
that's the reason. Okay. Each NADH has enough energy to make 3 ATP. So if I have 10 of them, that's why it's 30, right? Because one of them makes 3, right?
I got it. I got it.
Yeah, yeah.
Depend on that. Yeah.
That's right. So this, like, as to like why is it 3 and why is it 2? Like I said, that we don't have to worry about it. You just have to trust me on that. Okay.
Yeah. Thanks.
Yeah. Where is my thing here? Okay, okay. So I am hopeful that everybody will get this right. It's very straightforward. Anyone else? Okay, well, most of you got it right. It is glucose. Glucose is the one that is being oxidized. That's the one we are ripping the electrons from. And the electrons are being transported to the ETC, right? How? By the energy carrier, right? By the energy carrier, the NAD plus and the FAD. Okay. Let's think about this one. I didn't tell you explicitly what the answer is, but you can think about what we've learned and, you know, fill in the most appropriate term. Okay. Thank you. It says NAD plus, which is one of the electron carriers, is blanked into NADH when it accepts electron. Accepting electron is gaining electron, right? So remember, oil rig reduction is gaining of electron. So this is reduced. Okay, reduced. So to put it in an appropriate sentence, we are basically reducing the NAD plus and the FAD in step 1, 2, 3. And when it gets to step 4, it's going to be oxidized back into FAD and NAD plus. Okay, because then, you know, the electron has to get off, right? Okay, any questions? I know I ask you any questions like every 5 minutes, but seriously, anything before we finish this up? Okay, well then, I think this is a good place to stop. You know, this is like all the kind of like the background information that is needed for the lecture. If you want, you can go through the PowerPoint slides yourself. I know it's not a lot of slides, like 9 slides. That's what we just did with the notes, but that sets up an important stage for the rest of the PowerPoint. Okay, and we're going to be doing this next Monday. Okay? So before you go, though, I want to show you what's coming your way. You know what? Actually, I changed my mind. I was just looking at the time. It's still kind of early. Maybe we can do a few more. So I remember you don't, because we're gonna be losing a class for the family day, right? So we should probably do a little bit more. Sorry for the flip-flopping. But hold on, let me just pull up the calendar here briefly. Okay. So we are week number 5 next week. We will have lessons on Monday and Wednesday. I'm hopeful that we're going to be able to finish the lesson on Monday and maybe wrapped up on Wednesday. And then there's no class on Family Day. Right? And then your test is going to be on the 21st. So I want to do a little bit of test review with you on the 14th as well. Okay, so make sure you take a look at this calendar and plan accordingly. All right. So maybe just 5 minutes break first, and then we'll do a little bit more when we come back. Okay. So let's resume at like 1:47. Okay. See you in a bit. Okay, let's continue our discussion here. And spend a little bit of time talking about how ATP are actually being produced. Okay, so we'll just continue with our note here. Two ways, two mechanisms, okay, which means two ways. Okay, two ways, two mechanisms of ATP production in cellular respiration. All right, number one, the name is super long, but I'll explain what that means, okay? First one is called substrate level phosphorylation. Okay, that's a big, scary looking word. We'll just call it SLP for short. Substrate level phosphorylation. Okay, so let's explain what that means first. To phosphorylate a molecule means to add a phosphate. So for example, ADP plus P, the phosphate, becomes ATP. So in this example, ADP is phosphorylated into ATP. That's what the word means. Okay, I did not make up the word. It's a very long word. I know it's not very nice, but here we are. Okay, we have to learn it. So the ADP picks up a P, it gets phosphorylated. Right? So how does it happen? Well, the one way of this happening is by substrate level phosphorylation. Okay, so what does that actually mean? In substrate level phosphorylation, a phosphate, the P, I'm just going to put in bracket here to remind you what that is. a phosphate, is transfer from a donor onto ADP. With the help of an enzyme. Okay, so let's draw it out and see what it looks like. Here is the enzyme. And then we have two active sites, right? They were called. We learned that before. So over here, we have a fuel molecule, a donor molecule rather. Donor. And this donor is like, hey, I got a phosphate. I don't mind giving it away. If you want to take it, take it. And then on the other hand here, we have ADP. We have ADP. And ADP, don't forget, already has 2 phosphate, but we need a third one to make it fully charged, right? So the enzyme here, you need an enzyme for any reaction in the body anyways. It's going to take the P from the donor and then put it onto ADP. So this transfer of phosphate, it's going to generate ATP. We call this substrate level phosphorylation. Okay. Substrate level phosphorylation. Only a small amount of ATPs are produced this way. Approximately 11% are produced this way. It happens in step 1 and 3. If you don't remember what the names of step 1 and 3 are, that's okay. We worry about later. But you know, the first 3 steps, right? It only happens in 1 and 3. And it's only a small amount of ATP that uses this method. So that was the first method, okay? Substrate level phosphorylation. The second method is called oxidative phosphorylation. Right? So this takes place at the ETC, step 4. Okay. And the high energy electrons captured by NAD, H, and FADH2 are used to create ATP. Most of the ATPs are created this way. Okay, we'll say about 90%. Any questions about these two methods? All right. So one more thing before we go. There's one more thing before we go. Let's take a closer look at glycolysis. Right, so step 1. Glycolysis. Right, so where does it take place? Anybody remember?
Cytoplasm.
Very good. Takes place in the cytoplasm. What is going to happen is we split glucose. Glucose has 6 carbon, right? C6, H12O6, 6 carbon. We split the glycolysis, split glucose into 2 pyruvate molecule. If you split a 6 carbon molecule into 2, each of them will have 3 carbons. Okay? 3 carbons. So glyco, you should know by now, means sugar, and lysis, you should also know from last class, is to break. So we are breaking the sugar here into 2. Right. So this thing actually has 10 steps in it. Don't worry, you don't have to memorize them. Okay, there are 10 steps. Step 1 to 5 within sorry, I guess this could be confusing. You can call it stage 1 if you want, like stage 1, 2, 3, 4. And within the glycolysis, there are like 10 steps. Okay, so like step 1 to 5, we call this energy investing phase. Okay, so you might have heard people say it takes money to make money, right? Like if you put money in the bank, the bank will pay you interest. So you make some money from your money, but you have to invest some money first, or they're not going to start paying you for nothing, right? So that's what it's like here. You have to invest a little bit of energy into glucose first, and then they are going to pay you back. So in the energy investing phase, 2 ATP are used to energize glucose, right? So that it is willing to react, right? You energize it, you make it unstable, and then it's gonna react. Okay, so that happens in the first 5 steps. We're not gonna go into the details. The key thing to know here is we are investing 2 ATPs. By the end of the 5th step, we have not generated energy. In fact, we have lost some energy. Okay, we are running at a loss at that moment. And then step 6 to 10 is called the energy yielding phase. Okay, you are producing energy now. Not a lot, but you know, a reasonable amount. You will have 4 ATPs produced by, okay, if you have been paying attention, is by substrate level phosphorylation. That's where you take it from a donor and then you transfer it onto an ADP, right? So you're going to make 4 ATPs this way. And in the process, you will release some high energy electrons. Electrons are released and captured by NAD+, creating 2 NADH. Can someone please tell me where is this gonna go?
The ECT.
That's right. This is gonna go to the ETC, where you will take the electron to make more ATP, right? That method is called oxidative phosphorylation, as opposed to substrate level phosphorylation. All right. So this is in the PowerPoint. I'm just gonna make the slide come over here. Okay, let's take a look here. We start with glucose, and the black circles are the carbons, 6 carbons, right? We invest 2 ATP, and they become ADP. Okay, so this is the energy investment phase, the energy investing phase, right? Same thing, just different wordings. And then afterwards, we're gonna split the glucose into the pyruvate, which is 3 carbon each. I know it's confusing because I call it pyruvate and here it says pyruvic acid. There is a subtle difference, but for us, it doesn't matter. It's the same thing, okay? 2-pyruvate. Okay, you don't have to copy this down, I'll post it online. Okay, you're gonna release electrons, which is why we generate the NADH. Okay, and then we're gonna create 4 ATP. So summary. Okay, the key products of glycolysis. Right. How many ATPs did we actually make if we consider like the ones that we've used up?
2.
That's right. So there are 2 net ATP. I know we make 4, right? Like 2 and 2 here is 4, but don't forget you actually pay a little bit at the beginning, right? So gross production is 4. Gross is like before you take away your expenses, right? Net is just 2. And then we also produce 2 NADH and 2-pyruvate. Right. So these 2 things is gonna go on to stage 2. Okay. So right now we only make 2 net ATP. How many are we supposed to make? Anybody remember the number I wrote at the beginning of the equation?
36.
36 is correct. Okay, so far we've only made 2. 34 more to go. Okay. All right, that's it for today. Thank you so much for coming. Good luck with the lab, and of course, spend some time to study for the quiz as well. I will see you all on Monday. Okay, have a good rest of your week. Bye for now.
Yes,
Thank you.
for the
you.
quiz, lecture 1 and 2 is not included.